Follow us on social media and please hit the subscribe button so you don't miss out on any updates from our ALS experts.
Okay. Another fine adventure, folks. Welcome. Hello to everybody. I'm McFinn Levere, and my heartfelt greetings go out to each of you. Thank you to our team and you for volunteering your time and your heart to help understand and deal with this terrible disease. We are using all the tools we can find to help us, help you live a doable lifestyle. We have physical, mental, and spiritual needs. Our open forum is a perfect place to bring these subjects up. I just want to say, folks, I had a cup of tea today. And you know, there's sometimes on a cup of tea, a little, what do you call that? A little your fortune. And my fortune said today, smiling is the most basic kind of piecework. So I just want to say, smiling is a chemical reaction that could change your attitude. So try it when you can. Lisa, it's over to you.
Thanks for that, McFinn. That's really good advice. Simple, but really good. So welcome, everyone. Thank you so much for joining us tonight for our ALS talk. Our guest tonight is Dr. Johnathan Cooper-Knock, and he's from the University of Sheffield. And he's going to be talking to us tonight about the discovery of a new ALS Risk Gene and what this tells us about the cause of ALS. So as most of you know, February 28th is Rare Disease Day, and there's over 6,000 people who are diagnosed each year with ALS. So our entire team has been affected by this. We've all lost loved ones, and we have some who are living with ALS. So we believe hope comes from knowledge. We like to provide you with access to the top experts to hear firsthand about their research, so you know exactly what's going on, what progress is being made towards ALS. And we also believe that biomarkers along with genetic biomarkers along with digital biomarkers are super important in ALS to help us understand the course, the prognosis, and to help get people diagnosed much earlier. So the goal of our citizen-led research, which will help accelerate treatments as the ultimate goal if we can do this. So collaboration is key, and we need work together as a community to solve this. So please sign up for our speech study. I'll put it in the chat. It's the world's largest patient-led undertaken speech study with the collaboration of major research institutions and pharma companies to find treatments and a cure for this debilitating disease. It only takes 10 minutes a week, and thousands of lives can be saved in the future. So anyone 18 and over can participate in our study. So the format for tonight, we're going to quickly open up with a sharing some information of our RFI NIH survey, and then we're going to have Dr. Cooper-Knock to present. And following that, we're going to have Q&A moderated by our student ambassadors, James Sagasar and Zoe Laji. So they're going to ask the questions to Dr. Cooper-Knock. So then, as McFinn mentioned, we have our open forum portion where talk about whatever is on your mind. It's a safe place. So now I would like to introduce, we have Lane Olaf and Bevan Jett. They're going to be telling us what we did for this survey for the NIH and what the responses were and what it means for everyone. So Lane is a person living with a slow progression of ALS, and he's worked in pharma and biotech for many, many years. And Bevan Jett has a background in occupational therapy, and she's part of our ALS, Everything ALS team. She also is a caregiver to her partner, Dave Warnock, who also has a slow progressing form of ALS. And he's part of our Everything ALS team. So Lane, let's start with you. You have the floor now.
Thank you, Lisa, and welcome everyone. Good evening for those of you on the East Coast. Good afternoon for those of you on the West Coast. To give you a little background before Bevan gets into the actual study itself, as most of you know, in the US, December was a great month for ALS. You can't say that often for ALS. December was a great month because of passage of Act for ALS. Many people were floating on that and did not notice January also became a great month for ALS also, in that the National Institutes of Health, NIH, which is involved in almost all the research in the US in one form or another, either supporting it financially or actively involved with, decided ALS was going to be a focus for 2022. And they have decided they're going to put together a strategic plan for ALS. They have done that for other neurological diseases, some larger than ALS, some smaller than ALS. But 2022 is the focus for ALS. And as that process is getting underway with NIH, they have brought in a department with NIH called the National Institutes of Neurological Disorders and Stroke, NINDS. So NINDS and NIH are going to be working together to put together this strategic plan. The first step in this strategic plan was in January of 2022, they announced an RFI. As Lisa indicated, RFI stands for Request for Information. And some of you on this meeting may have submitted information for this RFI. So people with ALS, researchers, practitioners, organizations, all have submitted comments to this RFI, and they were to be submitted by February 11th of this past month. And with that, Everything ALS did submit a document indicating what is needed in the way of ALS research. And with that, I'll turn it over to Bevan to talk about what Everything ALS actually submitted.
To get our response, we wanted to do a survey because although we thought we had a really good idea, we wanted to have that backed up with data from actual people in the ALS community. Okay, basically we sent out a survey. We got 319 respondents back. We did ask who the respondents were because we weren't sure if we were going to be getting primarily people that were currently with AIDS or researchers or whatever. So here you can see that 46% of our respondents did have ALS. With PLS and PMA, because there had been a lot of discussion about how the new funding through Act for ALS, how that could have also affect people that may not have the classic ALS. So I was curious how many people were responding to that. We also have 11% caregivers, 21% who didn't necessarily identify as a caregiver, but were definitely a family member, friend, or ALS advocate. In our next slide, just again, trying to find out about ALS and what type. Not unexpectedly, 70% said they had sporadic, 16.7% said they had familial, and then we had almost 13% say they didn't know. I think that points a lot to the fact that not everyone's getting genetic testing for ALS, so they just don't know, and that's something we can discuss in the future. Basically, when we got results back, and what we were looking for is what were the most important areas for research and support that people were interested in. Over half of our respondents, the number one thing was promoting collaborations and partnerships between research facilities and pharmacological corporations and ALS advocacy organizations to accelerate discovery of interventions for the diagnosis, treatment, management, and prevention or cure of ALS. We were really happy to see this because this pretty much falls in line with what we've been doing with everything ALS. The other, again, remember this is going to equal more than 100% because we asked people what were their top 3 areas. Another 50% responded that this was improving the pathways from getting basically drugs out of the labs and into approved clinical trials. Other responses included basically researching for better technology, adaptive devices or drugs for managing ALS symptoms. 45% of the respondents wanted to improve how ALS clinical trials were performed to provide more equity and compassion to the participants, and also in just yielding better data, more measurable data for determining the benefit of a trial drug or treatment. And again, this is where everything ALS is very focused on as far as getting biomarkers, and that way we would be able to have more significant measurable data in determining the benefit. And again, there was a lot of different areas, and I just wanted to include a few more of the areas. A number wanted, people were interested as far as studying really basically what's causing ALS or what causes it to progress. That would be the genetic, molecular, cellular, or neural pathway changes. And then also really addressing just the caregiving, the barriers, and improving access for people living with ALS to be able to participate in the research process. Then another 26% identifying the biggest challenges on how to address for people currently living with ALS and their caregivers, which we actually asked at the end of our survey, and I'll give you those results. And for people who like visuals, this kind of gives you a really quick idea. Our number 1 area was the collaboration. Our number 2 area was getting treatments out of the labs and into trials so we can see if they work. And then we really kind of had a dead even tie between providing adaptive devices or drugs for managing ALS symptoms and improving how the clinical trials were performed. And this is the type of information that we sent to the NIH in our report. So in summary, what we got from over 300 surveys was that fostering collaboration among the ALS research facilities, missing a word there, drug discovery and advocacy communities, and improving pathways from discovery, research, and approval were really the big key things that people were interested in seeing happen. The other thing that we were looking at was basically what are the top 3 challenges for people living with ALS and their caregivers. Certainly there's plenty of them, but we wanted to find out what was really the biggest ones. And overwhelmingly, it was basically home health, finding caregiver support, and then financial and navigating insurance, and that's including Medicare. Further down the line was also as far as identifying and purchasing medical equipment, getting accessible housing, and also training and using the medical adaptive equipment. And I put together a little visual as well. Again, what really stands out is the fact of just the caretaking needs and the financial needs of ALS patients and their caregivers and family members. We did do because we basically did this survey by a checklist, so we may not have included things that people were interested in. So we did have an open-ended question, which basically you hand wrote in or you typed in, what do you feel is the biggest need, problem, or gap currently in ALS research? I thought it was very interesting. The big chunk of response was access to clinical trials and access or just even the availability of expanded access programs. That just overwhelmingly was our responses. As you can see, there's other responses that, it's just been the complaint about not having enough funding, not understanding exactly how ALS works as far as the stratification, so that basically no one drug's not going to fit all, and yet it seems like our trials are being conducted that way. So again, that kind of fell into how our ALS trials are conducted. And then again, that the FDA approval process and the trial process is just painstakingly slow for ALS patients. And that's it. Just kind of put a little word cloud together of all the things that came up in our survey.
Okay, thank you so much, Lane and Bevan, for sharing that with everybody. And if you want more details, we can put some information in the chat. But let's get to tonight's presenter, who is in London right now. It is past midnight, so thank you so much, Dr. Johnathan Cooper-Knock, for your patience. So Dr. Cooper-Knock is, like I had mentioned earlier, is from the University of Sheffield, and his research is focused around genetic causes of ALS. In particular, his focus is on the discovery of novel genetic mutations, which facilitate the design of cell and animal models for identification of new potential therapeutic targets. There was a paper that was just recently published on genome-wide identification of ALS collaboration, along with Dr. Schneider from Stanford Medicine and many others. So we'll post that paper in the chat. So please join me in welcoming Dr. Johnathan Cooper-Knock. Thank you for coming back to our platform to share your latest research. He was here in August, so we're excited to have him back again. So floor is yours.
Thanks, Lisa. Thanks. And yeah, great to be back. Really enjoyed hearing the discussion. I'll try and talk, I'll go a little bit off topic, but feel free to interrupt. I'll try and respond to some of the things that were said. Good. So can you guys see my screen? Is it all working all right? Yes.
Yes, perfect.
Yeah, fine.
Perfect.
So I'm glad your surveys talked about collaboration, and you want to see collaboration. I'm really behind you. I think you hit the nail on the head there. You know, we cut a lot of corners when we can join strengths rather than everybody trying to duplicate stuff. So, you know, I'm massive, and this whole project is a long-standing collaboration between my institute in Sheffield and Mike Schneider's team in Stanford. So yeah, like I can't agree more, basically. So what I'm going to talk about is, you know, so like I said, so I'm a clinician, I should say this as well. So I'm a neurologist, but my research is focused on genetics. So I'm going to talk about sort of our discovery of new genetic causes of ALS. And I'll talk a bit about why I think that's important, and where that can hopefully jump us into in terms of clinical trials. Because again, what I'm hearing from you guys, that you agree with, you know, access to clinical trials, getting these things into the clinic, absolutely key. I couldn't agree more. So I'll talk a bit about how this research is taking us towards that. But then also, I'll try and highlight what, you know, our sort of top hit from all of this work was a gene called KANK1. And in some ways, I think, you know, it changes a little bit on how I think about ALS as a process. And I think that has implications as well for sort of design of treatments. So when I talked to you before in August, I was talking about environmental factors that can sort of affect your risk of ALS. So I talked a little bit about exercise, I talked a bit about isoleucine, I talked a little bit about vitamin B12. And you sort of think, well, okay, so why is this guy's coming talking about one thing and then he's talking about another thing? It seems a bit like I'm all over the place. But the whole focus of my research really is this gene environment interaction, you know, leading to ALS. So if I think, so it's really interesting hearing what you guys are saying about, you know, you take a trial, you do a clinical trial, and you give a drug to all these different people, and they're all different, and we sort of expect it to work for everybody. And you know, some people seem to do really well, and then some people maybe don't, they're not affected, and yet we throw the drug out and then just say, okay, well, it didn't work, move on to the next one. And maybe it was an unrealistic starting point. I couldn't agree more. The way I see it is that we have to find what, you know, if everybody with ALS I see as being a specific interaction between a background genotype, genetics, and their own environment. And so we have to work out which interactions are important in which people, because that then allows us to say, well, this person needs this treatment and this person needs this treatment. Does that make sense? So then you know, it's personalized medicine really. And we are starting to see this. So many of you'll know, you know, for the C9orf72 genetic variant, there are now, you know, genetic trials aimed specifically at that genetic cause of ALS. So you know, Biogen, Wave Pharmaceuticals of another one who, you know, in fact, I was with a patient on the Wave trial with C9 today. And then, you know, for SOD1, we're also seeing this. So we are starting to see this, but I'd like to expand that from just being a few specific genetic causes to all patients. You know, if you could walk into your clinic and you could say, okay, well, these things are probably important from your environment. These are important from your genetics, you know, put it all in a little table and say, well, this is where we should go. That's kind of my vision. But focusing on the genetics, so the past genetic studies of ALS have sort of, you know, you've already mentioned, some of you, sort of familial ALS, you know, it's about 10% of all ALS, and that's, you know, that's kind of one gene, one disease. But then there's this 90% of disease that's kind of, you know, no family history necessarily, and you know, many of you may be in that group. But even those people, you know, who don't have a family history, we know that genetics is still important. So this is actually a plot from a paper from 2016 looking at the genetics of what we call non-inherited ALS. And it turns out, well, it is inherited. There are lots of genetic factors. It's just not kind of, you know, it's not sort of autosomal dominant, as in one gene, one disease. You have genetic risk factors, and some people who have them get disease, some people don't. It's a bit more complex. And these studies, what they do is they draw this line here and they say, okay, everything above the line is significant, but you can see below the line, it's not flat. There's a lot of signal here. Yeah. But we throw all this out because, you know, statistically we're underpowered. But the result is that we end up, you know, saying that we end up sort of assigning a genetic risk factor to maybe only 10%, if we're lucky, of people with ALS. Whereas what this is telling us is that most people with ALS have some kind of genetic risk factor. So, you know, we're missing all of this. So this project set out to try and get at some of this. And the way we tried to do it is we said, okay, well, this is the whole genome. And we know that the whole genome, you know, ALS is a disease of motor neurons. We know that motor neurons don't use the whole genome. They use the bits of the genome that are useful for being a motor neuron. So what we set out to do is find out which bits of the genome are important for being a motor neuron, throw out everything else, and then we can sort of, you know, if you reduce your search space, you find things a lot quicker, don't you? So the idea was that we improve our statistical power and find more genetic risk genes, and we did. We went from like 10 to 690 genes. So this is just reinforcing my point. So this is just saying that, you know, people, even if you don't have a family history of ALS, so this was a twin study, but looking only at people who have no other family history of ALS. And based on these twins, because twins share their genetics and their environment, they were able to estimate that sort of 61% of the risk of ALS, even if you have no family history, is genetic. So what does that mean? It's quite hard to sort of think about that. The way I think about it is that if everybody's walking around and they've got a number on their head that's their risk of getting ALS, 61% of that number is determined by their genetics and 40% is their environment. So, and if the two come together, then you either cross the line and get ALS or you don't, but you have that risk, and more than half of that risk is genetic. So genetics is important for all of us. That's the message I'm trying to get here. So the motor neurons we looked at. So we studied these motor neurons. We characterized them using a few different techniques, which I'll talk about in a minute. I had a very boring picture of a motor neuron to show you, but then Kesin, who I work with, she's an artist based at Stanford. So she put this picture together for an art competition using our motor neurons. So I thought it's far prettier than the pictures that I get off the microscope. So anyway, but these are the motor neurons. These are the motor neurons we studied. And this is what we used. We looked at these motor neurons and we said, which parts of the genome are you using? Everything else we'll throw out, because we believe that the risk of ALS is determined by, you know, is important within motor neurons. So if motor neurons aren't using it, it's probably not important. And so this just outlines the kind of the pattern of what we did. So we took the genetic risk here. So this is the picture I showed you before with the whole of the genome. And then these are the assays we used. We used things called TAC-seq, ChIP-seq, RNA-seq, doesn't really matter. But what we're doing here is looking within the genome of the motor neuron, finding the bits that are useful. And then we sort of use that to kind of look at the whole genome. And we were able to throw out 90% of the genome. So only about 10% is being used by a motor neuron. And so by doing that, we could go from those little peaks I showed you before, about 10 genes, to 690 genes. And then the whole of the rest of our work was kind of showing that these 690 genes were definitely important. So we looked in mouse models, we looked in tissues, we looked in other motor neurons, We looked at function, and I'll show you some of this data in a minute. We sort of looked at this within neurons. We grew up in a dish from patients and things. So showing that these neurons are, these genes, these 690 genes we've discovered are important risk genes for ALS. This shows how the model works. Now so RefMap it's a machine learning model we used to do this. It was designed by Sai Zhang, who's a postdoc in Mike's lab. I just want to credit Sai because he designed this machine learning model from scratch. So, but this shows how it works. So this is the score you get from the genetics. So this is kind of traditional genetic approaches. And every peak is signal. And I hope the impression you get is that it's noisy. Yeah, there's a lot of peaks. It's very hard. You look at that and you think, well, it could be anywhere, couldn't it? So, you know, it's hard to sort of, you know, spot the wood from the trees. But if you line it up with the things that are important within a motor neuron, you can see really it's only this region that is important in a motor neuron. And what are you doing? You know, there's quite a lot of genetic signal there as well. And so RefMap, the model, then says, okay, ding, ding, this is important, highlights this region. And it just so happens that this region is just downstream of a gene called ataxin-2, which you may have heard of because it's an important ALS gene. So this, you know, shows that how the model works, but also shows that the model is discovering things that we would expect. You'd expect ataxin-2 to be identified because we know it's an ALS gene. And like I say, we got 690 genes, and there were a lot of known ALS genes in amongst that 690. That was a highly significant enrichment statistically. And then we looked at it some other ways as well. So we took the 690 genes and we said, well, you know, can you, you know, if this is the biology that's important for ALS, then maybe we can ask some other questions. So what we did here is we looked at about 6,000 ALS patients where we had whole genome sequencing data. This is from Project Mine. And we said, how many mutations do people have in these genes? And we found that people had up to 5 mutations in the 600 genes. But it turns out if you've got 5 mutations, you develop disease at an earlier age than if you only have sort of 1 or 2 mutations. So that's what this graph is showing, that your age of onset is earlier if you have more mutations. And what that says to me is age of onset is different from risk of ALS. So it's a completely different thing. The only thing that links them is they're both in an ALS patient. So it shows me that we're capturing something of the underlying biology here, that these genes are not just plucked out of thin air, that they are actually capturing the thing that's driving the disease. So that was kind of, you know, like a reassuring thing that we are on the right track. And then because we've got this big list of genes, you know, whereas in the past it's been, you know, people have gone gene by gene and it's one thing at a time, and you can learn things from individual genes. But you know, with 690 genes, you can start to ask things about entire systems. You know, what is the driving force? It's like I think of it like an architecture compared to a brick. You know, a brick tells you some things, an architecture tells you the structure of a house. So when we looked at the 690 genes, we could start to say, you know, what is the, you know, so important thing about genetics is it's, you know, genetics is determined at conception. So it's fixed, and that means it's not affected by disease. It affects disease. It's upstream, not downstream, if that makes sense. So what is the upstream architecture of ALS? And it turns out that it very much highlights the sort of, if you think of a motor neuron, it's got this kind of cell body, this axon, and then the synapse where it joins onto the muscle or another motor neuron. And it highlights this bit, really, not the cell body, but it highlights the kind of the axon and the synapse and says that this is where disease is starting. And this is something I'll come back to, because I think, you know, this, there's some other reasons I think this is important. And I think this has implications for therapy going forwards. So we look, what about, you know, I've told you I'm going to talk about KANK1. So where did KANK1 come out of these 690 genes? Well, it was kind of the top gene in kind of the ranking. You know, everything has a ranking. In terms of genetic signal, it was the most highly enriched with genetic signal. But also when we kind of looked at it in a different way. So RefMap uses what we call common genetic variants. And by common, I mean, you know, they're present in sort of more than 1% of the population. So, you know, they're risk factors, they're not sort of causes of disease on their own, they're risk factors. But also we can look at rare things that might be more deleterious and might have higher effect in the same genes. And when we did that, we found that KANK1 had bad changes that were associated with disease, And then we highlighted these 3 genes because these 2 had some more bad changes, things that gave you higher risk of disease. But VAV2, mutations in VAV2 appeared to be protective. So people who had mutations in VAV2 appeared to be less likely to get ALS than the general population. So that was interesting. And then we sort of, as a validation of this, we jumped to a different data set. And here we're not looking at genetic mutations here. We're looking at level of expression within motor neurons. And this is the Answer ALS project. You guys might have come across this, but these are motor neurons that have been derived from people's skin cells. So about 1,000 people have donated, patients with ALS donated their skin cells to Answer ALS. Some guys at Answer ALS have developed these into motor neurons. And then once they're motor neurons, you can look at the expression of every gene in the genome within these motor neurons. And it turns out for the 3 bad ones that we identified with based on their mutations, the higher expression gives you earlier age of onset. But for the one that was good, as in it was mutated in people who were not getting ALS, then lower expression was correlated with an earlier age of onset. So 2 different measures, different aspects of disease, but all telling the same story, that more of these genes is bad, and more of this gene is, mutations in these genes are bad, and mutations in this gene is good. And these are all amongst our 690 genes. Okay, and then this was just a further validation. Again, we're always looking for confidence. So we looked at this completely independent data set with about 3,000 ALS patients and about 7,000 controls, 8,000 controls. And again, KANK1 was very significantly associated with ALS. So, giving us more confidence in that finding. So what do we do next? So what we did now is we sort of moved into, we wanted to see, well, what happens if we recreate some of these mutations in neurons? Do they get disease? And if they do get disease, ultimately what we'd like to do is then try and, trial therapies on these neurons. But we started with these neurons called SHSY5Ys. So they're not a motor neuron, they're a human neuron, but they're actually a cancer cell line, but they're very easy to manipulate. So they're a kind of a good starting point. They're not gold standard for ALS, but they're a good starting point. And what we did is we edited into these neurons, used CRISPR. We edited in the mutations that we saw in patients associated with KANK1, some different types of mutations. And what do you know, the neurons, they had toxicity, and more to the point, we saw their axons started dying. So exactly what I was saying before, that it appeared the disease was specifically affecting the axons and the synapses. And that's just shown here. So we did it for 2 of the genes, KANK1 and BNC2. And again, so the mutations that we CRISPR'd in, they reduced expression of the genes, but they also reduced the length of the neurites. So like the axon and the branches, and they reduced the length of any branches as well. So we're getting to, and they also killed the neurons. So we're getting toxicity that's in these neurons carrying the same mutations that we see in patients. And we're seeing kind of that toxicity is specific to the kind of the axon bit of the neuron. So then we moved into kind of the gold standard model for ALS at the moment in terms of cells in a dish. So these are iPSC derived, so they're derived from skin cells, but they're stem cells. You take the skin cells, you make the skin cells into stem cells, and then you make the stem cells into motor neurons. But these are quite a good model of ALS, and we could do quite a lot here to sort of test and see whether these neurons actually looked like the neurons you'd find in an ALS patient. So again, we used CRISPR. We recreated in these neurons the mutations that we found in KANK1 patients, and we looked at the survival, and we showed that the neurons there was toxicity. We also used a technique called patch clamping, so we could look at the electrical function in the neurons. And then we also used this, we looked at TDP43, and I'll talk more about TDP43 because this is really, really key to what makes a neuron have ALS or not. Is TDP is almost the definition. So this is just showing the toxicity. I can answer more questions if you want, but this just shows that the neurons, so these are the edited neurons, and then we had lots of controls. So these are the same neurons, but without a mutation edited in, and these are the same neurons, but with an edit in a different place in the genome selected at random. And you can see that it's the ones with the KANK1 mutation that have the increased toxicity, same in this graph here. So in terms of the electrical function, so this is patch clamping. This is done by a guy called Matt Livesey, who works in my institute. So, or not my institute, the institute I work in. So this is, I mean, it's a fantastic technique, but it allows, what Matt's able to do is to record from individual neurons in a dish. So he's recording how excitable a neuron is, and it's one cell at a time. So it takes a long time, but it's a very detailed technique. But what you see here is that the black is the kind of the mutations that the neurons that have mutations in KANK1, and they're hypoexcitable. So when you inject current, you don't get a response compared to these control neurons that don't have the KANK1 mutations. And again, we use lots of controls. We use mutations in other genes as controls, but it's the KANK1 mutations that cause the loss of that make these hypoexcitable. And that's interesting because that mirrors sort of things that have been seen in ALS previously, and suggests, you know, what it's suggesting is that these neurons really do have ALS. And then the kind of the proof of the pudding was this. So TDP43, many of you may be familiar with this, but I apologize if you've heard this before, but I'll go over it because I think it's important. So this is a normal motor neuron from spinal cord of somebody who doesn't have ALS. The brown is staining this protein TDP43. You can see the majority of TDP43 sat in the nucleus, but there is some in the cytoplasm. So just to orientate you, this is the cell body of the neuron. This is the axon and the dendrites coming out. You see it's a big cell, but there's a bit of TDP43 in the cell body, but majority is in the nucleus. It's very brown. When you have motor neuron disease, this gets lost. So this is the same part of the cell here. This is the nucleus, and the nucleus has lost its TDP43. The brown's gone. And what do you know? It's formed into this compact inclusion. So the TDP43 it sits in the kind of cell body in the cytoplasm, either as a compact inclusion, or you get these skein-like things that look very spidery sat in the cytoplasm. And this is 98% of people with ALS have this. If you look at their motor neurons, this is what you find. So it's almost like it's far more specific than, say, you know, people talk about amyloid for Alzheimer's. It's only about 75% of people with Alzheimer's have proper amyloid pathology. TDP43 is more than 98% of ALS patients. Very, very specific. And more than that, it actually correlates with neuronal death. So if you look in the brain of somebody with ALS, the number of these inclusions you see tells you, if you count the number of inclusions, you can predict how many neurons will have died. So that tells you that it's actually fundamental to the disease process. It's not just an innocent bystander. So you could argue if you could reverse this process, you'd reverse the neuronal death. But when we looked in our KANK1 neurons, we saw TDP mislocalization. So we saw TDP in the control neurons was in the nucleus. In the mutated neurons, it was gone out of the nucleus. The nucleus is blue here. And then again, it's formed into this compact inclusion in the cytoplasm, exactly what we see in the patient cells. And that's quantified here. So what we're saying here is that when we edit KANK1, this new gene, we see neurons that have ALS effectively. And that really gave us a lot of confidence in terms of our findings. But there's a bit more you can tell from, this just says that when you move TDP43 out of the nucleus, it has certain consequences. One of them is that you lose a gene called STAFMIN2, a protein called STAFMIN2. I won't talk more about that for now, but this was happening in these neurons. But what I think, what I wanted to highlight to you guys, because I think what this sort of says, so KANK1, you know, I guess the question comes, what does KANK1 do normally? You know, what's it doing in a normal motor neuron? Well, it's right here in the end of the neuron. It controls actin polymerization. So actin in here is in the green. It's right in the sort of the end of the neuron at the synapse. And so KANK1 is acting right down here. And what our experiments have shown is that when you mutate KANK1, you end up with TDP43 being mislocalized all the way up here in the cell body. Now, ALS research at the moment, there's a lot of focus on this part of the cell. But I think what we're arguing is that, you know, the KANK1 mutation is driving the whole thing. And we know that because the only thing that was different about these neurons was the KANK1 mutation. So we know that the problem started here and ended up here. And so what we argue in our paper, and what, you know, my thinking going forwards is that, you know, therapeutically, we need to think about this part of the cell. And if we sort this, then this will be sort of benefited. And that's kind of where I'm thinking at the moment. So this is just to highlight this really, axon degeneration. It's not like this is a totally new idea. I think what we added here in this paper is that we did it with a whole genetic architecture and made this point, quite forcefully. Whereas in the past, it's kind of been case by case. But these are old papers sort of talking about axon degeneration in motor neuron disease and is neuromuscular junction. So this is the kind of the synapse between the motor neuron and the muscle. And this paper from 2012 is saying that we should protect that part of the cell. So this is not a new idea, but I think what the reason I want to highlight these dates, I think things have moved away from this a bit, and actually a lot of work, you guys may have had talks, have focused on other things that are more in the nucleus, like RNA processing, DNA damage. Now, I'm not saying these things are not important, but I do wonder whether if the primary problems in the axon, maybe we're only correcting something that's downstream. Now, you may have a talk next week and somebody will disagree with me, and I'm very happy to be disagreed with. But I guess from our work, this is where my thinking's going. Good. So yeah, so that's what I had to say. So this is just to say thank you to all the people I work with in Sheffield, Pam, who's our head of institute, and Laura, who did a lot of the cell work particularly, and then Mike, who I believe is going to come and talk to you in a few weeks' time. So you'll see more of Mike. So he leads the lab in Stanford, and this is very much collaboration with Mike and Sai. So Sai works in Mike's lab, and this is just some of their previous work. So they are, they're very much genetics, but I approached them a couple of years ago, and since then they've been working on ALS, more and more. But yeah, very grateful to them. And then the other collaborators that be involved in this work, and Ice Bucket Challenge photos. But yeah, thank you. I'll stop sharing, but very happy to answer any questions. I hope I explained that well enough.
Thank you so much. That was very thorough, and we're going to dive right into the questions. And we are having Dr. Michael Schneider coming to present, but not until I think May. But that will be, we're looking forward to having him as well, and he can talk more in depth about other parts of the study. So we have James Sagasar from our student ambassador to help us moderate the questions, and I'm not sure if Zoe is here, but I
think she is. She just needs to
here.
be made into a co-host.
Great. Okay. So I'm sharing the doc with you guys, or James, I just shared it with you. So let's get going on the first one, and I'll let you guys take it away.
Yeah, thanks, Lisa. So first of all, I just wanted to say, Dr. Cooper-Knock, I thoroughly enjoyed your presentation. It was very informative, and I did not know that any of that had been like, I just figured like C9 ORF72 and SOD1 are just basically the not the only, but the main familial causes. And even like aside from the familial causes, I thought that there were really no genetic parts of the disease. And another thing that was really interesting is that I'm actually a neuroscience major in college, and I just learned about patch clamping in my class. So when you mentioned that, I was like, oh my God, I know what that is. So that was super cool. Okay, let's jump right in. The first question that we got was asking whether you have any suggestions about how to get involved in whole genome sequencing trials that might help to identify new genetic markers.
Yeah, great question. So increasingly there's kind of coming together of whole genome sequencing data sets, because I think really what people realize, numbers are power, and so the more numbers we can get, the better we'll be able to characterize these things. Now, so Project Mine is a global effort, and is the biggest whole genome sequencing effort for any disease, not just ALS, it's any disease, any single disease. Project Mine is the biggest. So if you ask your neurologist about Project Mine, they will have heard of it, and they will be able to, I tell you what, ask your neurologist. If they're not sure how to do it, then send me an email. I'm very happy for you to circulate my email, and I'll figure it out with you.
Very kind of you. I'm sure we'll be able to put your email into the chat somewhere.
I'll put it in.
Awesome. Well, our next question is going to be is, what do you think the time frame is for CRISPR and ALS?
Yeah, great question. So yes, you're right, because there's the obvious question here is, if I can CRISPR it into a neuron, can I CRISPR it out? And this has already been done for C9, and people have taken cells from people with C9 mutations and CRISPRed out the mutation. So I think CRISPR as a therapy, we're not quite there. So it's being used in the cancer field, but what they're doing is they're taking cells out of people, CRISPRing them in a dish, and then putting them back. The problem with using CRISPR as a drug, as you come in, I CRISPR you, and is off-target effects, and there's this, that we know that CRISPR is not pure, and that people will have off-target effects, and if one of those off-target effects could end up being more toxic than the mutation in the first place. So it's not quite at a state. It's getting there, and I use CRISPR predominantly. In my lab, we use CRISPR as a tool for modeling ALS. CRISPR as a therapy is gaining a lot of interest, and it is getting there as a therapy for all sorts of things. I mean, you can use CRISPR to cut out HIV, for example, from the genome. So because the HIV virus incorporates itself into the host genome, so it's got masses of potential for lots of diseases, but we're not quite there yet in terms of safety, basically.
Well, that's something we definitely can look forward to. So the next question is whether the gene research that you conducted included people that reverse from ALS, like McFinn.
Yeah, great question. So the answer is I don't know. So we included, we used lots of different data sets, probably getting on for about 15,000 different patients over all the data I showed you. It's, within those, there's likely to be some ALS reversal cases, but unfortunately, I don't know which are which. And unfortunately, at the moment, Finn, you're in a small group, and my goodness, we want to see more people like you. But I don't. Yes, there are. When you're dealing with big numbers, there's always a kind of a loss of detail. It is a problem, but you sort of work on the basis that kind of things that are real stack up. If you see something that's kind of rare, but then you keep seeing it, then you believe it's real. So it's that repetition that's power.
Piggybacking right off of that, this next question goes right into that. The hypothesis that ALS reversals have a genetic component in common that protects them from the disease. Could the method that you used also be used to discover whether that hypothesis is true or not, and potentially identify that specific gene?
Yes, but you need a lot of reversal cases. It's all about power, or you've got to shrink the search space so small that you've got enough power. Do you see what I mean? So it's the same argument where we reduce the search space from motor neurons, we're able to discover more genes. If you're getting numbers of ALS reversals, then you're going to struggle to get more than tens or hundreds, tens even. But you'd have to almost have to know where you're looking before you start. And maybe we'll get there. Maybe we'll get there. Maybe we'll have like a set of genes that we think is important for reversals, and then we can study the reversal cases and find the mutation. Because you're dead right. Like, it's a fascinating idea.
Okay, so the next question is, if someone had genetic testing that showed no genetic markers and no familial causes, does that mean that they could possibly still have a gene that was passed down and maybe epigenetics caused it to turn into ALS?
Yeah, great question. So I think it's this idea of it's genetic risk versus genetic cause, I think, is the key thing here. So whereas we know most of most of what we think about as ALS genetics, when C9, SOD1, etc., is a genetic. It's like one gene, one mutation, one disease. And I call it monogenic. One gene is the cause. It's 100% of the cause. There's nothing else, or very little else. Whereas what I'm talking about here really is kind of genetic risk. So we probably, everybody, if you take 1,000 people in the population, maybe sort of a few of them will have ALS, but some of the genetic risk factors may be carried by lots of those people, but it's not, do you see what I mean? It's a risk factor for the disease, but it causes the disease in combination with other things. I hope that answers the question. I kind of waffled a bit there.
I think so. Speaking of which, can KANK1 and or TDP43 be used as a biomarker for ALS, and is that even feasible in clinical practice?
Yeah, great question. So KANK1, I don't know yet. Certainly something we're looking at, in terms of expression. All right. TDP43, because of what I told you about it in the 98, so KANK1 is mutated in a proportion of patients. TDP43 is dysfunctional in almost everybody. And there are lots of efforts to kind of try and measure it as a biomarker. So far, we've not managed it as a field yet. The biomarkers that are progressing is, you've probably come across neurofilament. That's being used now to guide clinical trials. So people who have no symptoms, but have rises in neurofilament and have a genetic mutation that's known to cause ALS are being entered into clinical trials and treated even before they have symptoms. So we are seeing biomarker driving therapy with neurofilament, but not with TDP43 yet, although I agree TDP43 would be the gold standard.
So, okay. Sorry. The next question is, how does one get tested for all of the ALS genes that you mentioned?
Right. So at the moment, not all of them have got into clinical testing yet. So the whole genome sequencing is the key. But there's once from genetic discovery into clinical genetic testing, so I work a bit with ClinGen. It's sort of a kind of a clinical based genetic testing body in the US. And there's a kind of a process. What happens, you get an initial paper published, and then there's a couple of years before things get validated and then become sort of a genetic test that is done in the clinic. So there's a bit of a process to get to that. However, so from things that are already clinical tests, so done by any neurologist in sort of these panels of kind of known genes. We did a study in Sheffield where we took 100 patients who walked through the door in a row. So we started at point A and we took the next 100 patients that walked in the door, and we tested not for these 690 genes, just for like known genes that have been around for a while. And even then, we were able to find a sort of a clinically relevant mutation in 20% of patients. So actually, when you start doing when you start doing the testing, you find 1 in 5 even before you get to the 690 that we've discovered. And what we're arguing for there really, and I think others in the field as well, is that genetic testing should be part of being diagnosed with ALS.
I just want to double check and make sure we're respectful of your time. I just wanted to see how you were doing on your end there.
I'm good. I'm very
good,
happy.
because we got a few more. The next one was going to be, how do you quantitatively measure genetic risk via odds ratio, and specifically, what is the KANK1 odds ratio?
Yeah, good question. So it's a hard one to answer. So we measure it in terms of heritability. The actual, in terms of having so the reason I'm struggling to answer it is because there's a lot of some of the mutations we looked at, we sort of quantified them as a group because they're not entirely independent of each other. And so I find it's not possible for me to give you sort of an odds ratio for a single mutation. What I will say though is, we are talking the minority of ALS patients carry a KANK1 mutation. So this is not like C9 where it's 10% of all patients. Not that scale. But I think when you get the 690 altogether, we're talking a genetic risk factor in over 50% of patients from that 690. But that's a genetic risk factor, so it's not the cause of disease on its own, although it is contributing to it. I hope that makes sense.
Yeah, I think that cleared it up. So the next question that we got was, what exactly causes genetic mutations?
Yeah, good question. So, good question. So genetic mutations, so in some way, so interesting. So C9orf72 is thought to be a random change that happened, but probably happened, say, 10,000 years ago in a Viking, so goes the theory, because everybody who has a C9 mutation has the same genetic background in that part of their genome. And so the way people have interpreted that is that that it's come from what we call a common founder. And by common founder, we mean one Viking, basically. And the reason I say it's a Viking is because the incidence of C9 goes down the further away you get from Scandinavia, where the Vikings came from. So the hypothesis is that it came from a Viking. So some mutations occur sort of, so all of us have mutations that have occurred in us by chance. And actually, all of us will have very, very rare mutations or even unique mutations that maybe only we have in the entire world. So there is this kind of rate of mutation that's happening all the time. And that's in many ways, that's a good thing, because it stops us all being vulnerable to the same diseases, for example. So mutations that can happen, they can be protective or they can be harmful. But the fact when you get an inbred population where there's less genetic variation, then we know that's not good, and the kind of the more diseases. So it sort of can help. It helps, sort of having a background rate of mutation is a way of overcoming disease as a population. So it's kind of does that make sense then? So it's like a normal process, but as a byproduct, it can create these very harmful mutations. Does that make sense?
Mm-hmm.
I think anybody who's taken a science class ever in their life understands that genetics can get really, really complicated very quickly. Which goes to the next question is, for a hypothetical, if an individual has been tested and finds out they do have one of the genes that you mentioned that could potentially cause ALS, does that necessarily mean that their child has a higher chance of getting ALS as well?
So very good question. Very good question. And I think what this gets down to is the fact that we know, and with something we know already, we know that if you have a relative with ALS, your risk of it, even if there's no other family history, if you have a relative with ALS, then your risk is higher than if you don't have a relative with ALS. Now, what's really important to say is you're still not likely to get ALS. So this is what we're talking about here is a family situation where you have. So say my father had ALS, I don't have ALS, nobody else in the family has ALS. My risk of ALS is higher than if my dad didn't have ALS, but it's still unlikely to happen. Does that make sense? So yes, these genetic risk factors, they increase the risk, but it's not like C9 where it can, or SOD1 where you can think of it as a 50% risk. Or 100% if you have a mutation. It's different to that.
Just to clarify what you just said, are you saying that any relative who may have ALS is going to pose the same risk factor chance as a direct parent to child line?
So two different things here. So if you forget parent to child, okay, so if you have any relative with ALS, then there is a higher than background risk, so higher than the general population risk of having ALS. Mm-hmm. Now, but you're still very unlikely to get ALS. Okay, so that's important. Now, parent to child, where you have the different situation is, say, SOD1, where your grandfather had ALS, your father had ALS, and then so that's kind of the. And that's the kind of the familial form, that's the monogenic form, and that's very different. Okay, what we're talking about is not that. We're saying that the risk is still very small, but it's higher than if you didn't have a relative with ALS. Does that make sense?
I understand. Thank you for clarifying that.
Yeah, no, it's important.
Yeah, that makes better sense.
So yeah, sorry if I've confused things. And again, like very much happy if anybody has questions afterwards. I know these are, it can be quite, when we start to think about risk, there's lots of questions come to mind. Very happy for anybody to get in touch.
Yeah, we will definitely, your email is in the chat, so hopefully we'll be able, and okay, sorry, the next question is, can stress be an environmental factor for ALS?
Yeah, good question. So I don't know. So again, like I said at the beginning, one of my interests is kind of the environment and my environmental factors leading to ALS. And what I'm trying to do, in fact, what I was doing today is to try and bring the two together. So how can we look at what environment interacts with what genetics? Stress is hard to measure. So how you, the key, the answer is I don't know. But I think the key to answering the question, you need a really good way of measuring it, and measuring it in a lot of people in a systematic way. So there are questionnaires, but I can't do 10,000 questionnaires very easily. Does that make sense? Like, and not every questionnaire works the same for everybody. So yeah, these are things I'm actively trying to look at, but where it can be difficult is when it's hard, when it's something that's very hard to measure. But yeah.
And I'm sure stress levels vary for each individual as well, which also probably makes it more complicated. I think this is going to be our last question. Through your presentation, I didn't know that had so many genes had been discovered to be potential ALS risk factors. So this question deals with that. And an individual had genetic testing and found the C9 or repeated 145. Do you think it would be worth for them to be tested for the other genes that you mentioned as well?
Yeah, very good point. So C9 is interesting. So 145 expand. So nobody. Okay, so C9 is a whole topic on its own. 145 repeats is a lot, and it's more than kind of the background. But I have patients who have 2,000 plus repeats. So now nobody knows the golden question in C9 is how much does repeat length affect risk, and what is, people talk about 30 repeats as a cutoff. 30 repeats comes from the fact that that's what we can measure. So it's very difficult to kind of be sure, what the relationship between length and risk. In terms of other genes, we know for a fact that C9 often associates with other genes. It often doesn't work alone. So my logic would be, yes, that these things could be important, that other genetic risk factors could be important. There were a number, 1 in 10 individuals in our study had a C9 expansion. Now what I don't know is, I don't know the overlap between KANK1 and C9. But yes, no, I do think that other risk factors are relevant with C9, and we know the fact that there are lots of individuals out there with a C9 expansion who never get ALS.
I'm being told we have room for one question left, so I'm going to have this last one be it. If an individual has a sibling and they both have the C9 gene, the sister has ALS and the other sibling has PPA, how is one gene able to do? I'm assuming the question is both.
Yeah, C9 again. C9 is a question on its own. So yeah, magic question. Nobody knows the answer. I think the relationship between FTD and ALS, or PPA and ALS, so some of the discussion before I talked was about sort of differences in severity, long, slow progressing, fast progressing. I think something that I, as a clinician working with ALS patients, I have a real hope in the fact that I see families where they have a genetic cause and yet have vastly different sort of severity. I can think of 3 siblings I knew. One person lived for 20 plus years, and then in another, their brother, it was very, very fast. But my hope is that we can flip one to the other. If these things are so variable, then there must be ways of changing them. I mean, we had I'll tell you one story to finish. So we had a guy who'd never ever heard. So he was a gentleman. He didn't have ALS. His son had ALS, and his brother had died of ALS. He didn't have ALS and he was in his 70s. His son got disease in his 30s. He didn't know anything about genetics, but he used to ring our nurses and just say, "You need my blood, you need my blood," because he knew that he was the only link between his brother and his son, and he knew that he was fine. So he knew whatever he had was protecting him against the disease, whereas his son had developed disease in his 30s. And I think, that is the hope is that we can, that variability, those kind of changing things, the fact that ALS is very variable as a disease means that we can make everybody slowly progressive if we figure out what's driving it.
Well, I think as we saw from your presentation, we're getting closer and closer there every day. Just wanted to thank you once again, Dr. Cooper-Knock, for coming in. With the questions that we didn't get to, we'll send them off to you in email as well. Wonderful presentation. Thank you so much for coming back as well. The next portion, you're more than welcome to stay for. You don't have to. It's our open forum where McFinn is going to run it for us. And thanks again.
Cool. I'm probably going to go to bed.
Yeah. Thank you so much. I feel a little bit more reassured based on what you told me about family members, because my brother passed away from ALS, my younger brother, and every day I feel like, okay, I felt a twitch, and I'm like, I have so much anxiety over it. So you made me feel just a little bit calmer that it's a higher chance, but still not great enough.
Yeah, absolutely.
To worry about it. So I'm going to sleep better tonight. Thank you.
Good. Thanks. Cheers. Thank you.
Appreciate all you do for us. Thanks again for coming again. Go to bed.
Thank you, Johnathan.
Bye, guys. Bye-bye.
What a pleasure.
Thank you. Great presentation.
Thank
You have it, folks, from across the pond, as they say. It's a world that's trying to take care of this problem, and there's people everywhere. So, and you know what? We're people, and we're right here, right now. So anybody have something that really juiced them from this conversation that they might like to share?
Well, our presentation was great, as they all are, but I did want to thank James and Zoe, our moderators today. I thought they were exceptional, and I wanted to thank Bevan and Lane for their early presentation. That too was so interesting, and I know a lot of hard work was put into that. So thank you very much.